Okay, welcome. This is Globins video number 1. Throughout this video, you should be able to describe the structure-function relationship of a heme with respect to, that's WRT, with respect to oxygen transport. Describe the structure-function relationship of a globin with respect to oxygen transport. Predict the effect of structural changes on function for the heme and or a globin. List the requirements for oxygen transport and define key vocabulary terms. Those will be highlighted throughout. Let's get started. Alright, so hopefully you've seen these pictures before. Last time we talked about fibrous proteins. This time we're talking about globular proteins. Again, these are water-soluble with a hydrophobic core. That means the very inside of these proteins is made up of your valines, phenylalanines, isoleucines, leucines, and the three-dimensional structure is driven by the hydrophobic effect. The outside is water-soluble, so you have all your polar positives, polar negatives, and polar neutral amino acids, making it soluble in water. These are globular proteins because they usually have a globalish type shape or spherical type of shape with, in comparison to the fibrous proteins, which are water insoluble and don't tend to fold into these globulary, blobby type shapes. So we're doing globular proteins from here until many videos. Okay, so let's start with the very basics here. So we're talking about oxygen transport, and it's a really beautiful story of myoglobin and hemoglobin, and I hope that you're not sick to death of hearing them, but it really does make a really beautiful story. It's a great example of protein structure and function, tertiary structure, quaternary structure, all sorts of wonderful biochemical topics. So, all right, question number 1. What happens to most of the oxygen that we consume? We are humans. What happens to the oxygen that you breathe in? A, oxygen you breathe in ends up as O's or oxygens in your protein. So this would be like the oxygen in serine. B, the oxygen we breathe is converted to water in our cells. Or C, the oxygen that we breathe is eventually converted to carbon dioxide in our cells. And most of you will pick this answer. That is incorrect. It is actually B. This is one of the biggest misconceptions in biology and biochemistry. Yes, O2 goes in, and yes, CO2 comes out as you breathe, but oxygen is converted to water in your cells. The food that you eat is converted to CO2, and then you exhale it out. All right, oxygen is important for ATP synthesis, a process which occurs in your mitochondrial cells, but before you can get the oxygen to the mitochondrial cells, you have to get the oxygen to the cell to begin with. So oxygen has to get to all of the cells in your body, and it's usually transported through the bloodstream. So this is a story about oxygen transport. Okay, so we're going to start with the super basics. It turns out oxygen is not, is this surprising to you? Not soluble, not very soluble in water, which is essentially what most of your bloodstream is made up of, is water. So oxygen itself is not soluble in water, which means, see here, it's right here, oxygen polarity, not good. Carbon dioxide, also polarity, we'll talk about that in later videos, also solubility, not good. So what that means is that O2 cannot just easily or efficiently travel through your bloodstream all by itself. Meaning it needs help. And this is a story about that help. Okay, there's a couple requirements for oxygen transport. All right, so here's some basic requirements. Okay, oxygen transport or binding has to be reversible. So whatever it attaches to, it has to also detach. So there has to be sort of this state where it's, I really should say, it's bound to its transporter. But then it can also be unbound or leave its transporter. Think about this as like if you get inside a taxi cab and it needs to take you somewhere, you want to be able to get back out of that taxi cab. Now, if you need the taxi cab again, you got to be able to get back in. So that's reversible. You come in and you come out, and it doesn't cost too much energy to do that. And two, in its transport, it cannot be chemically altered. So when oxygen arrives at its destination, it still needs to be O2. It can't have an extra carbon on it, it can't have a hydrogen on it, can't have been chemically altered, can't add any extra atoms, can't take away any atoms. When it gets to where it needs to go, it still needs to be O2. So these are the requirements. Its transport has to be reversible. Again, think of this as a taxi cab. It needs to be able to get in and out of its transporter. And once it gets there, it has to stay the same. It has to still be O2 when it gets there. All right, so this is part one of a 4-part story about how oxygen is reversibly transported throughout the bloodstream without being destroyed. Okay, so those are the 2 requirements for oxygen transport. It has to be reversible, cannot be destroyed. And part 1, which is this video, is the wonders of the heme. All right, so first things first, oxygen is not soluble in water, which means we have to bind it to something that it can be bound to. And it turns out that oxygen binds iron 2 really well. So iron plus oxygen is happy. So iron binds oxygen 2 really well, actually too well. Iron 2, just this guy right here, plus an oxygen, is irreversible. Oh, sad. We just broke one of our rules. But, I don't want to say we, but iron 2 can be structurally manipulated to be more, or to have more reversible binding, to have a more reversible binding. Okay, so iron binds oxygen really well, too well, way too well for it to be reversible. So iron 2 plus oxygen is irreversible. We can alter iron in a way that we can increase the reversibility. Hopefully that makes sense. So it turns out that iron 2 plus, and all the irons have 6 coordinate covalent sites. So I'm going to do 1, 2, 3, 4. These are all in the plane, so an X and Y plane. 5 and 6, so those would be in plane Z. So it has 6 coordinate covalent sites. If we tie up 5 of these, leaving the last one open to O2, then this will increase the reversibility. Why can't I? I just can never spell, can I? Come back. And this one increases reversibility. Close enough. Increases reversibility of O2 binding. Okay, so we need to cover up or hide 5 out of the 6 sites. Now, 4 of the sites are going to be, the 4 that are in this plane here are going to be covered up by the porphyrin ring. Okay, and that is this structure right here is the porphyrin, so we're looking at it from the top down. There's some really interesting features of this, but basically each of these 4 nitrogens is going to covalently bond to the hydrogen, to the iron. Okay, so all of these dashes here are coordinate covalent bonds. Pretty strong. Coordinate covalent bonds. So each nitrogen can take up 1, and we have 4 nitrogens. So that will tie up 4 of our 6 spots. So the porphyrin ring plus an iron is what we call a heme. You have to have two of those things. If you take out the iron, it's only a porphyrin ring. If you add the iron, it's what we call is a heme. Vocab word. Vocab. Heme. Vocab. Porphyrin. You should be able to recognize the structure of a porphyrin, and you should be able to recognize the structure of an iron. I'm sorry, you should be able an iron. Well, yeah, okay, that one too. You should be able to recognize the structure of a porphyrin, and you should be able to recognize the structure of a heme. You should also see that the heme itself is a flat structure. The porphyrin is also flat. All right, so that leaves site number 5. If we call this 1, 2, 3, 4, 5, 6 has to be the last open site for oxygen. So I'm going to go into this picture right here. So you can see the iron is in the center. Maybe you can't see that. The iron here is in the center. Blue again is nitrogen. So there are 4. This guy right here is O2. There's the blobby for O. And then it turns out the site down here, the 5th site, which I call the 5th site, is occupied by, I don't know if you recognize this, but this is a histidine. And we call this the proximal histidine. Proximal histidine. And let's talk next about where does this proximal histidine even come from? So again, we need something that will bind oxygen because it is not soluble in water. It turns out iron binds oxygen real well. Too well. So well that it's irreversible. But we can manipulate iron a little bit to increase the reversibility. And to do that, we need to hide 5 of the 6 coordinate covalent sites on an iron. 4 of them are hidden with the porphyrin ring that creates the heme. And the 5th one is hidden by the proximal histidine, which leaves site 6 open to O2. Now, where the heck does the histidine come from? All right, so the histidine comes from proteins called globins. Okay? This is a vocab word. Globins. What do globins do? Globins are responsible for transporting oxygen. Now, some of them transport oxygen. Well, actually, let's just say this. Different globins transport oxygen in different ways. So myoglobin is abundant inside of muscle cells, and it transports oxygen from the outside of the cell. MB is the abbreviation for myoglobin. It transports oxygen from the outside of cells all the way into the mitochondria, where it is converted into water, and that helps make ATP. Hemoglobin is in red blood cells, and this is your primary transporter, or i.e. the taxi. The taxi that we're really talking about, transporter throughout the body or in the bloodstream. Okay, so globins are where the proximal histidine comes from. And globins are proteins, they're globular proteins that transport oxygen. Okay, so this is a question. Here's another question. So why put the heme in a protein? Like, why do we have to add the heme to the protein? Why can't we add it to a lipid, a polysaccharide, a DNA, or an RNA? Well, the reason we put it in a protein is because these proteins are globular, which makes them water soluble and helps, water soluble, which is essential for transport throughout the body. Lipids are insoluble. Polysaccharides don't form structures that are capable of transporting things throughout the body, and DNA and RNA rarely ever leave the inside of a cell and are not good. DNA doesn't have much other function other than storage of information, and RNA really just relates that information next. Okay, so that's why the heme adds to a protein instead of a lipid polysaccharide or DNA. But why does a protein even need the heme in the first place? Well, it turns out the amino acids, side chains of the protein, do not bind oxygen at all. So you have to have the heme group, and when a protein needs a prosthetic group, so the heme is a prosthetic group, this is a vocab right here, that makes the protein plus prosthetic is called a holoprotein. This is all one word, holoprotein. You might also think of it as the protein is now whole or complete, the holoprotein. If the protein doesn't have the prosthetic, so protein minus prosthetic, it's called apoprotein. Apo means without. Okay, so here's some other vocab words. Holoprotein, apoprotein, prosthetic. So the protein or the globins need the heme because the amino acid side chains won't bind oxygen in any way, and definitely not reversibly. Okay, continuing on with the story. So now we have our heme, which is attached to, which we said has two open coordinate covalent sites. One is for oxygen, one is for the distal histidine. So then your next question to consider is, how is the heme associated with the globin? Okay, so we already said that this is the heme right here. This is the site that's open for oxygen binding, and this is the proximal histidine. Okay, it's in close proximity to the iron. It's in close proximity to the iron. So there's two main ways that the heme is associated with the globin. The first way is that this part of the heme is nonpolar. Hopefully that you can see it's mostly all CH2s. And this part right here is polar. So this heme slides, the nonpolar end slides in so that the nonpolar region of the heme is interacting with the hydrophobic core of the protein. So the nonpolar region interacts with the hydrophobic core, and this polar region is sticking out and interacts with water. Okay? This also lines up the iron to interact with this proximal histidine through a coordinate covalent site. So this nonpolar region of the heme forms van der Waals interactions with the hydrophobic core of the protein. The polar region makes favorable interactions with water, okay? And then the proximal histidine forms a coordinate covalent bond. Okay, and that is how the heme is associated with the protein. So you need all three pieces. You need the coordinate covalent bond with the proximal histidine. You have favorable interactions with water because this polar part is sticking out. And then you have favorable interactions with the hydrophobic core. So that's how the prosthetic group sticks inside of the globin subunit. And then the next question is, how does oxygen bind to the holoprotein? So it binds in two ways. So one, we make a coordinate covalent bond between O2 and the heme. This is very strong. And then the next one is there is a hydrogen bond between the distal histidine, meaning it is distant from the heme, and the oxygen. That's how the oxygen is stabilized within the protein. Okay, so we've kind of gone over the wonders of the heme, or what I call the wonders of the heme, and how it's associated, and how so far we have increased the reversibility yikes, of oxygen binding with the heme plus the protein. All right, so sadly, this is not reversible enough. So the heme plus the protein in myoglobin is still not reversible enough for transport. Maybe I should say transport and delivery throughout the body. I'm getting close, but there's a final piece of the puzzle, and that is actually going to be the very next video. So I'll see you then. Bye.